we're going to do homework first. I did my homework last night. It was actually kind of fun. I know, I'm strange like that. It's okay. I enjoy doing genetics homework. There must be something wrong with me, right? So I already have it in a PowerPoint. I just built it on the other one. The other thing I did, but I didn't turn it on per se for you guys yet, is the stuff that we started talking about, like product rule and binomial expansion and stuff like that. So in Connect, they want you to work out the whole problem. I don't want you guys doing that. So I kind of broke the problems into pieces. And so in order to do that, the next homework is actually in Canvas. So it's chapter, where is it? Oh, it's a quiz. Okay. It's chapter 2, part 2. So this is actually in Canvas. This one's more like an actual quiz where you can only open it once and then it's going to tell you the answers. Okay. But it's not hard. I broke it into pieces and it's multiple choice. Okay. 10 questions, 1 point apiece. Try to get that done before Tuesday's class. So what we're going to do now is, as I said, I put the rest of the homework and what I wanted you guys to read over in a PowerPoint. So we're going to go over that first, and then we'll finish up chapter 2, hopefully. So the fork line method, I told you to look at the sample. So they say for heterozygous for 3 genes, right? So in this case, this would be a huge Punnett square, right? Forget how many squares that would be. It's a lot. So instead of doing that, you can do the multiplication method they show you, which, I don't know, it just doesn't jive for me. But like I said, this isn't necessarily math class, right? The more important thing is that you understand how these things are done, or could be done. And just like, you know, the new math, like Common Core, and people are losing their minds about it, it doesn't matter really how you do the problem, as long as you get the correct answer, right? So if you want to write a big, huge Punnett square, which I'm not going to ask you to do, you could do that. If you want to do the multiplication method that they show you, you could do that if you know how to do it. Or you could do fork line if you know how to do that. Make sense? Right? So, you know, some of the math they're teaching kids nowadays is like counting by tens, or, you know, regrouping things, and y'all, it's whatever makes sense to you. When I was a kid, I did this all on my own. No one taught me this. When I add 5 plus 8, I actually add the 2 5s together and make 10, and then I realize, I mean, this seems really long, right? But for some reason, this is the way my brain works. There's 2 left from 7 and 3 left from 8. So it's 15. Like, that's how my brain groups numbers. It works for me, right? It doesn't really matter how you do it, though, as long as whatever method you're doing it correctly and you get the correct answer. That's the beauty of math, though. There's always a correct answer, right? Unlike science, right, there isn't necessarily always a correct answer. When you're dealing with mathematical science like we are, right, you can get correct answers, right? But then, of course, there's always the exceptions to the rules, right, like Miss Chimera, right? So I added that link for you guys, too. I wanted to remember to tell you that when I was still in Canvas. So I decided to just go ahead and put it here with our outline lectures and modules. So I put that link right here, a link to an interesting story that one of your classmates shared. All right, so that's that link right there. So I like fork line. It makes sense to me. And I'm actually going to use it to solve one of our problems later. So you would end up with 8 different male and 8 different female gametes possible if you're looking at 3 different traits for heterozygous individuals for each one of the 3 traits. So you'd end up with all of these possibilities, right? So yeah, 64 squares. Wow, that'd be insane. Draw that big of a Punnett square. So the alternative, right, is multiplying by each other. Especially if you're just doing phenotypes, right, it kind of narrows it down. But the first thing you want to do is just take each trait, and we can do this, right? We can look at each trait separately. So I have the Punnett squares, right? And we know this ratio, right? For 2 heterozygous, you're going to end up with 3 dominant and 1 recessive, which is true for no matter what trait for this simple system that we're looking at right now. So the 2 possibilities for tall or dwarfed, right, is a 3 to 4 or 1 to 4. And then your next line, right, are the 2 possibilities for the next trait. But you have to do that for each of the possibilities, right? So the lines keep forking, hence the fork line method. Right, so that you get all the way to the end, right, and you have all the different probabilities for the different phenotypes that you would see. Now, if we were to do genotypes, this would be much more, right? So, the question for the homework number 6, it says a plant produces wrinkled green seeds was mated to a plant that produces phenotypically round and yellow seeds. In the F1 generation, half of the parents produced round yellow seeds and the other half produced round green seeds. What was the phenotype of the parental plant that produced the round yellow seeds? So what I do is I just start from the beginning, right? And I start writing right next to each one of these what the possible genotypes could be. So we know for wrinkled it has to be little r, right? Because that's the recessive trait. They've got to be homozygous recessive. But for green, that's true too. It has to be little y, and we're using y because the dominant trait for color is actually yellow, right? So when we get to this one, this is why this one's in question, is if you're round, you can be either homozygous dominant or heterozygous. And then for the yellow as well, you could be homozygous or heterozygous. Based on the cross, we know what they are for each of these traits, right? So let's look at round first. So all their children from these 2 parents, the wrinkled green and the round yellow, are all round. So what does that tell you about the genotype for this round parent? We didn't get any wrinkled, right? Because we've got to get 2 little r's for wrinkled. So he doesn't have a little r probably, right? He's 2 big r's. He or she. Well, they're plants, so yeah. So big R, big R. Now let's look at the next, which is the color. So we are getting yellow and green. We're getting the recessive. And remember, green is the recessive. So it's little y, little y. Where did this guy get the little y? Well, obviously from this first parent, but it also had to get it from the second parent. So the second parent is heterozygous for the seed color. Everyone still with me? So what's our answer? What we have highlighted, right? Big R, homozygous, and heterozygous for yellow. But then the question goes further, right? So we know, so the round, did I mess this up? We're allowed to self-fertilize. Did I mess that up? Oh, damn it. Thought I was doing so good. I think I missed something. So it should be big R, right? Big R. I don't know if that somehow that got messed up. Okay. So these parents are allowed to self-fertilize, right? So the RR with the heterozygous Y were planted, and those were allowed to self-fertilize. So they're crossing them with themselves, right? So big R with homozygous with heterozygous for that trait. So did I write them out right? Oh, see, it's wrong on here too. Here, I thought I did my homework right, and I screwed up. Okay. So this guy crossed with this guy. Yeah, I got the right answer before. I think I'm messing up somewhere.
Yeah,
I think I am right that this, I'm just not.
I think it's big R, little r, because the original parent was
Right.
big R.
I'm skipping a step.
Yeah.
I did have it right. Because I was like, I got the right answer. What am I doing? Why am I changing it? I lost track of where I'm at. Okay. So, oh, here, this is why I messed up. Here we are, right here. So in the F1 generation, half of the plants produce this. So these guys are, we figured out, are R little r and Y little y seeds, and the other half produce this. The round yellow ones were planted, and those plants were allowed to self-fertilize. In the F2 generation, what fraction of the seeds are genotypically identical to the plants of the parent generation? So that's way back up here. So first we have to figure out, right, so this cross, and this is two heterozyguses, right? I didn't screw it up. Okay. But we're looking at genotypes, right? So we have to write out the percentages for the genotypes. So if you have heterozygous individuals, and I changed that, didn't I? Let me change it back. You have homozygous 1/4 of the time, 2/4 or half the time you've got the heterozygous, and then you have 1/4 recessive. The same for both of these. Still with me? Okay. So, instead of drawing out, right, big, huge Punnett square of 16 squares, I did fork line. Okay? Because it'd actually be more than 16. I forget how many it'd be. So, yeah, I rewrote it here, right? If we do the two heterozygous, these are the genotypes we get. We do the heterozygous for this trait, this is the genotypes. No, I did multiplication. I didn't do fork line yet. Okay. So we want to find out what percentage are going to be like the very first parent generation, which we figured out to be homozygous recessive, homozygous dominant, and heterozygous, right? These are the grandparents, or the parental first ones. Everyone still with me? So now let's figure out what percentage is going to be this, based on these genotypic ratios. So what percentage of them are going to be RR? 1/4, right? And then what percentage is going to be YY? 1/4 as well, right? So a quarter is 1/16. Still with me? Now for the next one, capital R, 1/4, right? But heterozygous for yellow is 2/4 or a half. So those times by each other is 2/16. The question asks how many are going to be this and this. So what do we do with these two numbers? We add them together, and we get 3/16. I would make that one less complicated on the test. But if we can do this for homework, right, the test will be like cakewalk. All right, question 8. So we want to cross wrinkled with yellow. So again, remember, wrinkled is going to be little r's, it's recessive, homozygous. Yellow can be one or of two possibilities, right, because it's the dominant trait. So seeds to the plant with round, again, round is the dominant, so it can be homozygous or heterozygous and have that phenotype. And then green is little y. So in the first generation, you observe all the progeny are round and yellow. So what are the genotypes of the parents if all their kids are round and yellow? So these are the ones we have to figure out. So which is it if they all ended up yellow? Is it homozygous or heterozygous? Homozygous. Because if they was heterozygous, we should have ended up with some green ones, right? But we didn't. So the same is true for the round, right? We ended up with all round. We didn't get any wrinkled, so he's also got to be homozygous. So with me?
So instead of like a number, they're just going to say they're always all yellow, like round and yellow. We just assume there's not like a little outlier or anything.
Right. The probability is, again, they're not telling you the sample size or anything like that, but they're, you know, in this case, we're staying pretty black and white.
Okay.
But yeah, you're right. There could be outliers depending on how big is the sample size, right? Maybe there's a sampling error, right? But based on the information, if all of them, really all of them, right, then we don't have any heterozygous for these traits, because then the recessive would show back up, right? Everyone understand his question and what we mean by that? Okay, good question. So the first parent, we know wrinkled, because that's recessive, right, is homozygous recessive. Based on the information we were given that all of the kids are yellow, then we know that this parent has to be homozygous, not heterozygous, for the dominant trait. And then for the next parent, as we said, it's round homozygous for the dominant trait. And because it has the recessive trait, we know it's YY. So, you know, that's more of a test-like question, you know, one step. Right? I'm not going to ask you multiple step questions on the test, and if I do, I'm going to separate them out individually so that if you get one part wrong, you're not going to get the whole thing wrong. Does that make sense to you guys? So I kind of did that too for the homework for this weekend with the binomial expansion and product rule. I did ask you guys some parts, but I separated them out. Right? So the question kind of repeats a couple of times, but I'm asking you a different thing each time. So remember, what 7 traits are we looking at? So are we looking at flower shape or flower color? Color. Pod color or pod length? Pod color. Width or shape of the pod? Shape. Seed size or seed shape? The seed number in the pod or seed color? Color. Height of the plant or length of the root? Height of the plant. Flower position or stem thickness? Flower position. And again, some of these, right, especially when you're doing stuff like length, even the height would be, you know, there's a huge difference with the height though, so they were able to do that one. But things like seed size, can you imagine like sitting and measuring? Yeah, it was like kind of insanity. And pod width, you know, it kind of makes sense why these weren't chosen, is what I'm trying to say, right? They weren't pretty discrete things that you could discern, where the color was, you know, 2 choices. And like we said, he got really lucky with all of these. They're all on different chromosomes. They're all actually loss of function genes, right? You either have it or you don't, and followed the standard, what we're used to, dominant recessive pattern. So as far as monohybrid crosses of two heterozygous individuals, when we're just looking at one trait, what is the phenotypic ratio that we see? If they're two heterozyguses that we cross for a single trait, phenotype, would you, yeah, 3 to 1. 3 dominants to 1 recessive. What law did this help demonstrate? Mendel's laws. There's 2 choices, right? Law of what and law of what? So this is where, right, you get 2 chromosomes, 1 from each parent, and randomly you get one or the other choice, right? When you separate and give to your kids. You separate and give to your kids. You need to know this stuff, y'all. Law of segregation, which states, right, that during gamete formation, when we make our gametes, we randomly pick between the chromosomes which ones we're going to give our kids. And we divide everything that we got from our parents in half to give to our kids. But you don't give, say, all the chromosomes you got from your mom to your children, right? You give a mixture, a random mixture that you got from either one of your parents to your children. For a dihybrid cross of 2 heterozygous individuals, what's the phenotypic ratio? 9, 3, 3, 1. 9 that are dominant, 3 that are dominant recessive, 3 that are recessive dominant, and 1 that's recessive for both traits. What law? What are we left with? Independent assortment. And this states, again, if they're on separate chromosomes, these 2 traits are not going to follow each other, right? They're going to segregate independently from each other. That's if they're on separate chromosomes, though, right? Stuff on the same chromosome obviously goes together. So I don't know if you guys have gotten a chance to look at it, but I'll put it up. I forget when, if it's been here a week or not. But that's a poster from the Human Genome Project, and it has on it, like, they've figured out where certain mutations are for certain diseases on the different chromosomes. And chromosome 4 has listed on it Huntington's disease. And one of your classmates in this class actually has her husband's parents. One of them had that. So she was asking me questions about it. And when I was picking out the homework, one of them was on Huntington's disease. I was like, sweet, I'm going to pick that one so that you guys will learn about it, and that'll answer all her questions when she does her homework. So that's one of the ones that I picked for the homework you'll be doing over the weekend. So that's it for the homework. And so let's go back to Chapter 2's lecture. And let's review for a moment. So 2 things, product rule and binomial expansion. So the product rule is the probability that 2 or more independent events will occur is equal to the product of their respective probabilities, right? Which gets kind of confusing. So what do they mean by independent? They mean that one does not affect the other, right? So being a boy or a girl, 50-50 chance, right? Because you're a boy or a girl doesn't determine whether you're going to have blue eyes or brown eyes, right? Those are two separate traits. Two separate probabilities. If you wanted to know the combination of the two, we do just that. We multiply the two probabilities together. Product, right, is multiplication. And again, it's not math class, so I'm going to ask you to figure out the individual probabilities in the homework. I'm not going to ask you to then multiply them together. So I don't care about that. I mean, you can plug that in on a calculator. But figuring out individual probabilities, I do expect you need to be able to do that. Does that make sense to you guys? So, and then given a problem, so you need to recognize, okay, for a particular problem, what do I do? Is it product? Is it two separate things, but I need to multiply them together? Or is it binomial expansion? So why would we use binomial expansion? We would use that, this crazy equation that, again, I'm not going to make you fill in, represents all the possibilities for a given set of unordered events. So can anyone think of an example of this where we would use this? So there's a set of events, and we want to know the probability within that set. So what do we mean by a set? What do you guys think? So a family could be a set, right? So my family, there's, 3 girls.
I'm looking at that estimate because we just actually did that. The X and the N would be considered 1. So it will be T.
Did that for what?
X. We just did it today. But we had numbers like P squared.
Are you talking about in math class?
Yeah.
Oh, okay. Because I was like, I don't know what you're talking about.
No, I was like.
Yeah. But don't worry about the equation or the math. We're talking about the principle.
Oh.
Right? This class, you don't have to stress about the numbers, right? We're going to talk about the principle of it, right? So why would we want to use this? This allows us to figure out the probability for a given set of unordered events. So what would be a given set? Say a family, right? What is the probability, right, that within a family of 3 that you're going to have girls, all girls, right? We could calculate that, right? It's not a very high probability, but yet look, I have 2 sisters. Does that make sense to you guys? So like say within a family of 7, what's the probability that all of them are going to be boys? Yeah, the probability is 50-50 with this, but when we fill in, if we were to fill in the math for this, which I'm not going to do, but here's one of the examples. So 2 heterozygous brown-eyed individuals have 5 children, right? What is the probability that 2 of the couple's 5 children will have blue eyes, right? So when you have a set like that and you want to know a particular question about that set. Does that make sense to you guys? Okay. So, and then you would calculate it out. I'm not going to make you guys calculate out binomial expansion. I just want you to be able to know when you would use it and then actually know the individual probabilities that you would then plug into the equation. Okay? So you'll practice that for homework. We'll go over the homework on Tuesday like we've been doing. Okay? So remember though, the homework's in Canvas, not Connect for this one because I broke it down to what I want you guys to know. So this is the last topic for Chapter 2. and this is chi squared.
That was pronounced chi squared, but that could be.
I've always said chi, but it could be chi. This is where this is a statistical mathematical equation again. So again, I'm not going to make you figure out the math. I'm going to tell you what the math is, and then you need to apply it to this. I didn't set up any of these. I was too tired by the time I got to this point last night. So none of these are in the homework yet. All the things were starting to run together. It was time to go to bed. I didn't want to screw it up. Okay, so we want to figure out the goodness of a fit for a hypothesis, right? So we start, we have a predicted hypothesis, right? Based on what we know about genetics, right? We can predict what the outcome of a cross would be. And then actually doing the experiment and getting the data, taking that data and seeing if it fits our hypotheses. We're doing this mathematically. Right? So this is a statistical test. It doesn't prove that a hypothesis is correct, right? It just helps support it, or definitely causes us sometimes to reject it. So this is the formula. Again, not one you're memorizing or calculating. Thank goodness. But so where we take our observed data, compare it to the expected data based on our hypotheses, all of that gets summed up. And the reason why it's called squared is because it's squared. So we have to take the square root. So looking at fruit flies, Drosophila, there's a gene for wing shape. Trouble saying that word. So for the genotype, they write it C+ for normal wings, just a C for curved wings. And then we could also look at body color. We have normal body color E+, which is gray, and E, which is ebony. The wild type allele is designated by the plus sign. So that's the most common in nature, what they mean by wild type. And it makes sense that it's the dominant one, right? Recessive is a mutation that happened, right? So it's denoted by the lowercase letters. So if we did a cross between two true breeding flies that are normal winged in gray and curved winged in ebony, the flies of the first generation are allowed to mate with each other to produce the second generation. So we end up with heterozygous for the first, right? Everyone still with me on that? And then the second generation is the heterozygous is mating with each other. So it's that big, huge 16 square, Punnett Square. So if we're looking at two traits, right? So that's a dihybrid cross, where the parents are heterozygous, what's the expected phenotypic ratio? This is one we should know. 9:3:3:1, right? So this is what they actually got, right? So straight wings is another word for normal. I don't know why they changed their terminology. These are the actual numbers that they got out of 352 total flies. So taking that data, as opposed to what we would predict, that 9331, times that by, right, so this is what we expect, 9 out of the 16, right, so for 300, oh my god, dyslexic moment, 352, we expect to get 198 of them that are straight-winged and gray bodies, right, and then so on and so forth. When we calculate that into the equation, expected versus actual, right, so observed versus expected, we get a number of, 1.06. Once you have that number, which I'll give you that number, I'm not going to have you guys calculate out this number. That's the probability. We need to look at the probability chart, which again, of course, you're not going to memorize. I'll give you the chart. Low chi-square values indicate a high probability that the observed deviations are due to random chance alone. A high chi-squared value indicates low probability that the observed deviations are due to random chance alone. So what do we mean by low and high? Typically, if it's 0.5, which is 5%, that's a low score, right? To use the chart, we got to figure out the number of degrees of freedom. That's calculated by the total number of categories minus 1. So in our experiment, that would be 4 minus 1, because there's 4 different things, right? The 2 different colors and the 2 different wing shapes. That's 4 characteristics, right? Minus 1 would give you 3 degrees of freedom. So when we look at this chart, we'd be looking at this row for degrees of freedom. Our value was 1.06, right? So it would be around 80% probability that that hypothesis is correct, right? That it isn't just random sample error. If we're over in this, right, you usually would not accept that hypothesis, right? You don't want to be, less than 5% match between your expected, your hypotheses, and what you actually observed for your experiment. But again, sample size can really affect this, right? You want to make sure you have a large enough sample, right, that you're not going to end up with a lot of variation. So, as we said, 0.8%, which is 80%. So it's quite probable that the deviations between the observed and the expected in the experiment can be explained by just random sampling error. Right? It's not that it's not a good fit. This is a good fit. So I'm going to put together some problems this weekend for you guys to do next week. Work on good fit, work on probability and binomial expansion. So I have 10 questions for you. It's actually like 3 or 4 questions broken up into pieces. And it's under the modules. Remember, we're skipping over Chapter 3 and going to Chapter 4 next. And I'll have that up for you guys hopefully by Friday, the new PowerPoint for the next one. So there it is right there, the link. It's right there in Canvas. It's not going to take you in to connect. And it will tell you the answers after you do it. What else did I want to... No, I just have it set up once. Just once. Later, I think I can put it so that you guys can take it again if you want, but I want you guys to just do it once. So.
Is it time?
No, it's not timed. I never put times on mine. I don't like that ticking clock thing either. I know what I wanted to do. I wanted to go... So the other thing I'm going to say is that I'm not going to assign any more crosses, right, or any more homework than I have. But if you struggled with the homework, right, and I mean pretty much once you do it and it tells you the answers in Connect, I think, too, and we went over them, too. So redoing them is not going to help you, right? So if you need more practice, that's something you should understand whether you know if you need or not. There are, in your book, right, more problems that you can do. The answers to those problems, if you're using the ebook, are right there, except, let's see, I am at the end of the chapter, yeah. So there's the solve problems, which they answer it right then for you, right? But then you have the concept questions. And notice it's every even one, they give you the answer. And so if you're looking at your physical book, by the way, it's Appendix B, where all the answers are written out in the back of your book. With the computer, you know, they just can make links. So you can just click. So there's these conceptual questions. And so, of course, like I said, you only have the answers to the even. But I'm the teacher, right? So I have the answers to the odd as well. So if you do any of the odd, right, and you want me to check it, you can either email it to me or bring it to class, and we can go over it. It's also a good time to like come by office hours or something like that and do Punnett Squares with me if you want, if you're struggling with it. Because I love doing them because I'm crazy like that. So I don't want to get started on 4 yet today. I think we've done enough today. I don't think anyone's going to disagree with me. Anyone disagree with me? Not today. Okay. So I'm going to let you out early. This is going to be one of those rare times. Just to make up for the really long, you know, introduction that we had. Which I'm actually going to grade introduction. So if you haven't introduced yourself to the class, do that. So I'm going to grade those and we can get to know each other better.